All right, so in this video, we're going to talk about now the process of taking the messenger RNA, which is going to copy, it's been transcribed, copied from the DNA, and taking that and leaving the nucleus, coming out to the cytoplasm, and building, right, the protein, amino acid, one amino acid at a time. So we do call that translation because we are translating from the language of nucleic acids to the language of proteins, right? And once we have that protein, we have the ability to do work. And remember too, for inheritance, that once we have the proteins are considered your traits, right? These are the things that are unique about us that allow our functions, give us structure and appearance. So this translation does occur, of course, in the cytoplasm. It's good. What does it require? Well, it's going to require enzymes. It's going to require messenger RNA. It's going to require transfer RNA, shown here, right, that moves amino acids. Free amino acids, the ribosome and ribosomal RNA are for a few things, right? So transfer RNA carries the amino acids. So this is right, it's literally transferring. So you, in your cells, you have these pools of free amino acids that basically come from either your diet or they come from the recycling of other proteins by lysosomes and other cells. And these transfer RNAs are specific to a certain amino acid, and they're going and picking it up in that cytoplasm, bringing it to the messenger RNA. Sorry, this is cut off a little bit, but translation then uses a codon, right? We said three nucleic acids, three nucleotides, uses a codon to determine the amino acid. So, and each codon is unique. So AUG is a methionine, whereas uracil is a phenylalanine, all right? And then you've got the cytosine, right, guanine, adenine is an arginine. So each one being unique, and that's how you build all these different proteins. So specific sequence starts to be built, all right? Again, as I've already said, those amino acids came from, right, your either recycling of other proteins in the cell through the lysosomes, or they can be taken up from the amino acids from the blood that came in through your diet, right? So here, this cartoon is really nice. It kind of shows everything all together. Here again, slightly different appearance to these transfer RNAs, but same concept. Ribosomes form along the messenger RNA at one end. It's always going to be the AUG, by the way. That is a start codon, and there has to be a marker to start it. And they start fitting, right? They use anticodons to make sure this is the exact amino acid we want. And since the anticodon is unique for the actual codon, you get an amino acid, a unique amino acid. So comes in and starts building. So the binding of this transfer RNA to mRNA occurs in association with the ribosomes. So the ribosomes allow all these things to come together. And then the ribosomes perform the enzyme function of synthesizing, attaching, forming the peptide bonds between amino acids. As I already said, it needs energy, it needs enzymes, free amino acids, messenger RNA, transfer RNA, and of course ribosomes. All right? So here's the big picture assembly line. We can make a lot of protein molecules in a very short period of time because of these processes. So here again, genome inside the nucleus, the DNA doesn't leave. Some sequence on that is known as a gene. This is being read by pulling apart, right, separating the two strands shown here, and building then a new molecule. And this is definitely an RNA because you see the uracil there. Right, this new molecule, single-stranded, is small enough to leave, and does leave through the pore, comes out and assembles with ribosomes, and the ribosome then has a binding site for the transfer RNA, which comes in and specifically binds to the codons on the mRNA, and the codon then, an anticodon relationship, signify that the amino acid is correct. This is the amino acid we want in this position. And it just moves along. The ribosome just kind of moves along, we say, reading this messenger RNA and translating it to a protein. All right? So it's pretty cool. All right, so the last part of this chapter looks at, okay, what do we do and how do we get new cells? How do we fulfill that function of growing and and producing more and more and more cells, right? Or producing cells when there's been damage to a cell, repair and replace, right? And this follows nicely on this process of transcription and translation, because the process of building a new cell, right, that we're going to talk about now, and results then in cell growth and producing more and more cell numbers, getting to that trillions, from one cell to trillions, requires us to replicate that entire DNA that we said we could do in 6 hours. So we really want to focus on this idea. And the replication of this DNA occurs here in this phase, collectively known as interphase. So again, growth, reproduction is a characteristic of life. And so the cell, right, now has to produce a copy, right? And this was how you went from one cell in the very beginning of your life to trillions. Cells are not immortal. They wear out, and some cells can be replaced. Generally, epithelial cells and connective tissue cells can be replaced. And you also can damage them in the course of your life, clearly. I'm sure you've damaged your cells at some point or another. And again, some cells can be replaced, some cannot. So typically, muscle cells and neural cells do not undergo, right, the mitotic phase anymore. So they cannot make copies of themselves anymore. They can't replace themselves when there's injury. So this is something to consider as well. What is it that controls? I say signals here, but controls, tells a cell it needs to reproduce. Well, there's a couple of really straightforward things, all right? When some of these are internal, right, and they are part of the cell, things like cyclins, specific proteins. Some of them are external, like growth hormone. Growth hormone, right, triggers cells to begin dividing. Cells also stop reproducing at certain points, and we use other proteins to signal that. And cells will also stop dividing when there's too many cells in a given area. They won't overgrow and become disorganized, because remember, organization was a characteristic of life. When the cells do not actively divide anymore, as I said for muscle and nerve, we say they are arrested in G naught. And that's where they'll stay. Right? Most of your life, and most of the cell's life specifically, is an interphase. Really, 75 to 90% of the time, when we look at this cycle, right, is interphase. And I just say business as usual. If you're a pancreatic cell, you are making and producing, right, transcribing, translating all these proteins that are enzymes to help you with digestion, right? So you grow, and so sometimes the G's are for, sometimes people call them growth or gaps. They used to be gaps, but really growth is a better term, cell growth, right? So the cells grow during interphase. They start manufacturing proteins, and they maintain the jobs that they're supposed to do, okay? If we're going to divide, right, then you have to replicate all that DNA, because you can't have a new cell if it doesn't have all the instructions. So if you are a cell that's going to divide, and we said epithelial or connective cells generally do this, then in the S phase, and think of S for synthesis, you're going to replicate or synthesize your DNA. And your organelles, right? You gotta create everything that's going to be useful to a new cell. And I say here, you now know that the chromosomes are packages of DNA, and you know how DNA replicates. We talked about that in a previous video. So that's really what you're doing here. And here it shows, again, these are homologous chromosomes. They kind of color code them. You got one of these chromosomes from your mom and one from your dad, and remember, you have 46 chromosomes, and chromosomes are a bunch of DNA. So this cartoon also shows that these chromosomes, in this case, have actually copied themselves. So they've gone through this S phase. So this was one of them, and it made a copy of itself. And we call these guys sister chromatids. And this was one, and it made a copy of itself. So these two blue ones are identical, and they're actually sister chromatids as well. All right? The process of dividing the nuclear material is called mitosis or karyokinesis, and it's divided in 4 stages, right? In this process, one cell will become 2, always. The 2 cells are identical, or clones, of what we call the parent cell. It will distribute the replicated, all this copied, synthesized DNA as chromosomes, and the organelles into 2 new cells, right? Because that's what growth is, add cell number. The 4 stages are prophase, metaphase, anaphase, and telophase. I often see this acronym PMAT on quizzes. And it looks a lot like this. So I say here, you just really need to know the main events of each stage or phase and know the cell structures, right? And these are all aspects pretty much of the cytoskeleton, centrioles and spindle fibers, microtubules. So they're involved in helping connect and move, right? Because the chromosomes are the packages. They're just going to sit there unless, right, moved around. So in prophase, we have this, condensing, right? Chromosomes are condensing from chromatin, and you can then begin to see them. Spindle fibers start to form, the nuclear envelope breaks down because you need to free up, you need to give this structure access to these structures, right? And the centrosome divides and starts to move to opposite sides. When you have centrosomes in the form of the centrioles on opposite sides, you then say you're prometaphase, but you don't have to separate this out, all right? And there's these structures called kinetochores that also appear, but again, not going to ask you about those. Metaphase, I always say M for middle, because at this point in prometaphase, we attached the spindle fibers to the chromosomes, and now we have moved those chromosomes right down the middle. So the original and their copies are lined up, and for you, this would be 46 chromosomes down. All right, anaphase, A away or apart, and these are actually showing the actual divisions here in cells that have been stained with fluorescence. And you're splitting, right, this centromere that's holding the original and its copy together, and you're moving each of these chromosomes to opposite sides. And then in telophase, I always say that's kind of the opposite of prophase. You're really reforming the nucleus, and you are now going to unwind these chromosomes. The centrioles are going to come apart, right? And during this time, you will also undergo this process called cytokinesis, in which the cytoplasm will squeeze apart. So around the middle of the cell, there'll be some actin filaments that align, and they just start, right, contracting, because actin is a contractile protein. All right, so here showing this cytokinesis, right, and again, this is a cell, the cytoplasm separation. Starts maybe in anaphase, late anaphase, continues through telophase, and then again, it's a contractile ring of actin that apportions or separates the organelles and the cell contents. So one cell is now officially 2. The mitosis had separated the nuclear material, so cytokinesis separates the cytoplasm. All right, so how do we control this? This is really key, isn't it? And you know what, I'm actually going to save this for another video, because this is a good place to stop. So we'll come back and talk about this in the next video.